Hello Brainerds, my name is Jason. Welcome to the Big Brain Busters. So in this video we are going to talk about the formation of the bilaminar and trilaminar germ layers. Okay, so if you remember from our previous video, we talked about formation of the blastocyst. We said that the blastocyst contains the inner cell mass, which we called the embryoblast, and the outer cell mass, which we called the trophoblast. Okay, so now after this process of blastocyst formation, which occurs in about 1 week from the day of fertilization, the next thing that occurs is simply formation of the bilaminar germ disc. So how the bilaminar germ disc occurs is how we are going to explain in this video. So next thing, one thing you should know is that these inner cell mass will start now differentiating. So as they differentiate, they will proliferate, and then when they proliferate, just here on their base, they will form a number of flat cells which will be lined to them, and these flat cells here are what we will call the hypoblast. So these flat cells which are coming from the inner cell mass is what we call the hypoblast. Then this inner cell mass will now differentiate, and then they will change their shape, and then instead of looking the way they are, they will now become columnar in shape. Okay, so this is how the hypoblast has been formed. The next thing what will happen is that these cells which have now become columnar in shape will attain the name epiblast. So formation of the flat cells known as the hypoblast and the columnar cells known as the epiblast is a process where the bilaminar germ disc has been formed. So let's look at the next process. So the next process is simply the formation of the amniotic cavity. So what will happen is that between the trophoblast and between the trophoblast and the epiblast, okay, there will be a space that will be formed. So how will this space be formed? There will be fluid invagination between the trophoblast and the epiblast, and then the trophoblastic cells will start proliferating, and then they will form amniogenic cells from the trophoblast. So these cells here are what we call the amniogenic cells. Then due to fluid invagination into this cavity here, we will form a cavity known as the amniotic cavity, which is filled with amniotic fluid. After formation of the amniotic cavity, you've seen this hypoplastic cell which will later on form the endoderm. So this hypoblast cell will now continue to proliferate, and then it will form another layer of flattened cells, or perhaps should I say rather another group of flattened cells that will cover this cavity known as the blastocyst cavity. They will be embedded in the blastocyst cavity. So this is how it will look like. So as you can see, the hypoblastic layer will now become like this, okay, being embedded in the blastocyst cavity, and then this membrane that has been formed, this membrane of flat cells, is now known as the Hauser's membrane. So this membrane here is the Hauser's membrane, and then the space that is in between this layer of flat endodermal cells is now known as the primary yolk sac. Okay, so now we are at the point where we have formed the primary yolk sac. So now the next thing that will happen, do you see this space that is in between the walls of the primary yolk sac and the trophoblastic walls, right? The trophoblast, this space here. This space will be occupied by proliferated trophoblastic cells. So the trophoblast will start proliferating, and then the cells will start invaginating into this space, and then they will form a structure known as the extraembryonic mesoderm. So this part here now that has been formed is known as the extraembryonic mesoderm. Now take a look at what has happened. Remember that the amniotic sac, or the walls of the amniotic sac, which is the amniogenic cells, were attached to the trophoblast, but then due to invagination of the trophoblastic cells, this amniotic sac, or the walls of the amniotic sac, have been separated from the trophoblast, and then to form the extraembryonic mesoderm. Okay, then another thing that will happen is that these flat cells of the primary yolk sac will turn into cuboidal. They were flat, but then they will become cuboidal. They'll change shape, and then when they change shape, this primary yolk sac will attain the name as the secondary yolk sac. The next thing that will happen is that there will be death of these cells which had invaginated. So there will be cell death, which is apoptosis of these cells. Okay, so as her cell death is occurring in this region where I am shading white, okay, this part here will join, these spaces that are being formed will join together. But then there is a limit. They won't reach this region. They will not reach this region here, where I've shaded, where I've put black there. So I will explain why. So what is happening is that as apoptosis is occurring in this region, they will form an important structure known as the chorionic cavity, or the extraembryonic coelom, this part here. Now take a look that they have not reached this part where I did not shade white. So why? Remember that this structure here is attached to the trophoblastic cells via this part here. So now if you put, if this invagination of cells reaches here, if this apoptosis reaches to this point, meaning that this guy won't have any point of attachment. So meaning that for us not to have such, we need to end somewhere. And then due to that, this is where now we have our future connecting stalk. So this part here is the connecting stalk of the of this structure, all right, of the bilaminar germ disc, this circular structure that we have formed. Okay, so this part here now is being attached to these trophoblastic cells by the by the connecting stalk. The next thing is that this extraembryonic coelom, what is happening is that it has divided the extraembryonic mesoderm into two parts. It has divided it into the into this part here, where I am shading black, this part. Okay, this part is between the walls of the secondary yolk sac and the walls of the and this space that has been formed, this extraembryonic coelom. So this space here is known as the visceral or splanchnopluric extraembryonic mesoderm. Okay, then it has also divided this space, this extraembryonic mesoderm, into this space here, the outer one. Okay, this space between the trophoblast and the extraembryonic coelom. Now it is called the parietal or somatopluric extraembryonic mesoderm. It is also known as the chorionic plate, because remember, a plate is where you place something. So meaning that this chorionic cavity, or the extraembryonic coelom, it is resting upon the chorionic plate. Okay, then the next thing also that has been formed is simply two important membranes, and those important membranes are the chorion and the amnion. So the chorion is the membrane that will be formed in between the in between the amniotic, sorry, the in between the chorionic cavity and the trophoblast. So this is the chorion. Then the amnion is the membrane that will be formed in between the amniotic cavity and the and the chorionic cavity. Right. Alright, so I hope we are together. We are from talking about formation of these structures here, which are the extraembryonic coelom and these other structures. And I also mentioned to say there is this part known as the connecting stalk, where this circular germ disc, I'm calling it circular because it is now circular in structure. So this circular germ disc is attaching via the connecting stalk to the trophoblast, to the trophoblastic cells. And it is this very connecting stalk that will form the future umbilical cord. So there will be degeneration of some cells, then it's going to form a tube where there will be formation of arteries and vessels, and it is in those arteries and vessels which will connect to the placenta, and then there will be, exchange of different substances such as important nutrients and also waste products. Okay, so with that being said, let's proceed. So now the next stage where I'm going to be describing is simply the formation of the, or in short, it is the process of gastrulation. Okay, gastrulation is just simply the process by which it is the process where the two germ layer, or the bilaminar germ disc, this bilaminar germ disc that contains the epiblast and the hypoblast, will be converted into a trilaminar disc, which contains the ectoderm, mesoderm, and the endoderm. So this process that I'm going to be describing is known as the gastrulation. Now it begins at this stage. Okay, it begins in the third week of gestation period. And what happens is this. So there is a very important structure that we need to view. So now this important structure that we are viewing, you cannot view it properly if you are viewing this circular germ disc in a longitudinal section, as we have been doing all along in this longitudinal section. We need to view it in a transverse section. We are viewing so this diagram here, we are viewing the circular germ disc from the top. Okay, it's more like we are looking down on it. We are, viewing it from the top. So to just make your understanding better, it is the same as what we did here. We have our columnar cells there, these columnar cells that I'm going to draw here. So these are the columnar cells that you've seen. So these are the ones that make up the topmost bilaminar layer, which is the epiblast. So these are the columnar cells that we've had all along. These are the columnar cells here. So these columnar cells are these guys here, these columnar cells. Then we have now the cuboidal cells that are down here. I hope it is making some sense, because I want you to get it at this point. So these are the what? These are the cuboidal cells, which are down here. Okay, the cuboidal cells, which are forming the what? The hypoblast. So these are the guys here, right here. Okay, so now then we know that on down here there is the what? There is the yolk sac. Then up there we have, up there we have the what? We have the amniotic sac. Okay, so I hope it is making sense. So now why we are viewing it from the top view, because this structure on top here, this round structure, okay, is what we want to see. And this structure here, it is noted when we notice that these cells that are of the hypoblast, which are cuboidal, they start turning their shape into columnar shape, and it occurs at this point towards the head end, because this structure now is elongated. In most of the cases you find that it looks something like this. So this is the head end, or the cephalic end, and this is the caudal end. Okay, this is the caudal end, or the tail end. So this is the same thing. Then this round structure that I've drawn here, you will put it there. Okay, then this round structure is known as the procaudal and it is the future mouth. It is what will turn into the mouth. Okay, then now it is via appearance of this structure which will notify us to say, oh, actually this is the center. Okay, this is the center. Without this appearance of this procaudal plate, we won't know which one is the center. So it is this very procaudal plate that divides our structure into the left and the right. Okay, so now in this case, since now we have our procaudal plate which has columnar cells around it, the next thing that we will notice is that there will be the next thing that will happen is that there will be formation of a primitive streak. So the primitive streak will start forming from the tail end, this side. So it will be first of all like an out bulging, an out round bulging, then it will start growing towards the center. Then as it is growing towards the center, it will pause right there. It will just pause just right there. It will end there. Then this part is what you will call the primitive node, and this part is what you will call the primitive streak. Okay, so now to show you better diagrams, I try to draw this, so don't mind my drawing. Okay, so this is what I meant. This is just simply an illustration of this, what I've already talked about. Don't get confused. Then I have put it so that we can view it on top like this. Okay, we are viewing it from the top view. Okay, so now in this case, I was saying that appearance of the procaudal plate will notify that we are now in the center. The procaudal plate is the structure that divides this embryo into right and left. Then I said that there will be an out bulging here, just right here. Okay, and then this out bulging, known as the primitive streak, will start growing towards the center, and then it will end right there, just at the center. Then there will be formation of this structure known as the primitive node. Okay, the primitive node. Then this structure here is the primitive what? Is the primitive streak. Take note, the layer on top is the what? Is the epiblast. The layer on top is the epiblast. Epi. Then the layer down is the hypoblast. Hypo means down. Huh? So now, this primitive node, it's more like an out bulging. Okay, it is more like an out bulging. Then this primitive streak is more like it was also an out bulging, but then it will become a depression, a small depression like that. So as you can see, there is a depression there. So now what are the functions of these guys? So gastrulation is simply the process by which this bilaminar germ disc is turned into the trilaminar germ disc, and this is the process of gastrulation that we are going to be explaining. So what is happening during gastrulation? So what is happening is that there are cells all over here, these cells on top of the epiblast. Okay, on top of the epiblast. So these cells here will do what? They will start invaginating, they'll start migrating from this part. So let me just draw this properly so that you understand. So there is this part here, this primitive streak that will end at the center and become something like that. So these cells of the primitive of the epiblast will start moving. They'll start moving in here. Let me use another color. They'll start moving, migrating from there into the primitive streak, entering in there. So what is causing the invagination of these cells from where they are into this structure? So what is causing that is simply a factor known as a fibroblast growth factor 8. So this FGF8 is the factor that is responsible for causing invagination of the epiblastic cells into the primitive streak. And as these cells are invaginating, first of all, they will go and replace the cells of the hypoblast. And then when they replace the cells of the hypoblast, this hypoblast will now be called the what? The endoderm. The endoderm. Okay. Then this endoderm now, it has been formed, right? Then continuously these cells of the epiblast will continue invaginating in here, but then because there is too much invagination of these cells of the epiblast, these cells now will start moving. They'll start moving laterally like this. Let me use another color, maybe this one. They'll start moving laterally, and what's this? They'll start moving laterally, and they'll start moving towards the head end. So they'll be moving laterally towards the head end, laterally towards the head end. So I hope it is making sense, because now there is too much invagination of these cells. They are entering through the primitive streak. Then, as they are doing that, they will form another layer in between here, another layer just here between this ectoderm and the endoderm. So the epiblast, sorry, because we haven't yet formed the ectoderm, but already we are almost there. So remember, we've already formed the endoderm, right? So due to invagination, in between this epiblast and the endoderm, there will be formation of another germ layer in between here due to continuous invagination of the cells. Then as this continuous invagination of the cells continues, there will be formation of the what? Of the mesoderm in the middle. So these cells now that have remained, those which belonged to the epiblast, will now become this layer now of cells that has remained on top will now be called what? It will now be called the ectoderm. Ecto. Instead of the epiblast, it will be now called the ectoderm. Okay, then this middle layer that has been formed due to continuous invagination of the cells into the primitive streak is now called the what? The mesoderm, from the word middle. Okay, then the layer that is down here, this red layer that is down here, that was formed due to replacement of the hypoblastic cells, will be now called the what? The endoderm, the innermost. Okay, so this gastrulation process starts occurring in the beginning of 3rd week, around day 15. This is when this process starts occurring. Okay, and I hope this is where gastrulation ends. So immediately you form your 3 germ layers, the trilaminar germ layer or trilaminar germ disc. This is what marks the end of gastrulation. So this whole process that we talked about, by formation of the primitive streak, formation of the procaudal plate, and formation of these whole germ layers that we talked about, this is all what we call gastrulation. Okay, so we proceed. Okay, so this is where we end our video. I hope you enjoyed learning with us on the processes of gastrulation, and I hope you are going to like, share, and subscribe to this video and the channel, and hoping that you will drop a comment in our comment box so that we may know what your concerns or what your questions are about anything medicine related. So this is where we end our video. Please enjoy your day. Shalom, shalom.